I'm happy to be here today with Dr. Robert Brown. He is Chair of Translational Oncology at Imperial College London, where he heads the Epigenetics unit and has a joint post with the Institute of Cancer Research. His abstract is Epigenetic Marks as Biomarkers and Their Mechanisms of Maintenance as Targets in Ovarian Cancer. His work is funded by the charities Cancer Research UK and Ovarian Cancer Action. Thank you for joining us.
Pleasure.
Would you discuss the importance of robust prognostic and predictive epigenetic changes as biomarkers and how they impact clinical outcome for ovarian cancer?
Okay, so ovarian cancer is a particularly frustrating cancer. It responds to a variety of treatments, surgery obviously, but also platinum-based and taxane-based chemotherapy. However, despite that, in the last 20 or 30 years, I think it's fair to say that we've not made huge improvements in the overall survival of ovarian cancer patients, despite being such a chemoresponsive disease. Now, that may be because we've not got the right drugs, or it might be because we're not using the drugs in the right way, or not putting them into the context of those existing successful therapies. So what biomarkers in general can potentially do is to try and stratify patients who will particularly benefit from particular treatments. Long-term aim, of course, would be something like personalized medicines. But being able to stratify patients that we know will respond to existing treatments will perhaps allow us to use the new therapies that are coming through in a much more rational way and really try and target them to the right patient populations. So we've been interested in epigenetic regulation for a number of years, and I think it's fair to say that compared to some of the genetic biomarkers that people are using, it's perhaps a bit behind the trend, shall we say. There's not been the same technologies available to be used. But the last 5-10 years has really seen an explosion in available technologies to look at epigenetic biomarkers, and in particular in this DNA methylation, this methylation of so-called CPG islands. These are kind of regions of the genome which are found near the promoters of many genes, so near the beginning of many genes, and are intimately involved in controlling the levels of expression of the gene. So whether a gene is on or off, it's really like a switch, on or off. And because these technologies are now available to allow us to measure this methylation, and not just measure it in tumour, but also to be able to measure it in easily accessible tissues, like tumour DNA being released into plasma, then we can start to begin to look at whether these DNA methylation biomarkers can be used to identify those patients which will particularly benefit from the existing treatments, and importantly, those ones that will not. So in the case of ovarian cancer, in particular in the case of high-grade serous ovarian cancer, sadly only some 30% of patients will be alive after 5 years. So we really want to identify those patients who will do badly, and then try and use some of the newer therapies that are coming through really much earlier in the patient journey.
Would you discuss the association you've discovered between methylation of multiple promoter CPG islands at genes in the Wnt pathways and progression-free survival of epithelial ovarian cancer patients?
Sure. Well, we've been working with clinical troops. As you can probably tell, I'm Scottish. And initially we've been working with a group called the Scottish Gynecological Clinical Trials Group, which despite its rather parochial name is actually a network of clinical investigators throughout the UK and Europe. And we've been doing various translational and biomarker studies. We've been collecting ovarian cancer samples, and we're very grateful to all the patients who've consented to allow those samples to be used for research. We've been collecting these samples for over 20 years now, so we've got really very good, mature clinical data. And so we examined that initial cohort for methylation, as you say, of these CPG islands, which are involved in controlling expression of genes in this cohort of tumour at this particular pathway called the Wnt pathway. And we know that the Wnt pathway is intimately involved in tissue homeostasis. It's involved in regulating the number of stem cells. And it's a very, very important pathway in controlling cell growth in a whole variety of tumour types. Not been studied too much in ovarian cancer, it's fair to say. So from that study, we first of all took an in silico, a bioinformatics approach to try and identify all those key control regions that were involved in the levels of expression of those genes. And then using a molecular platform called differential methylation hybridization, which is a microarray approach, which basically looked to ask the question whether these CpG islands are methylated or not. In other words, whether they've got 5-methylcytosine, which we know switches genes off when it's clustered at these islands. So we looked in the cohort samples that had been collected through the Scottish Gynae group and saw a very significant association between methylation of certain loci and the length of time till patient's tumour would come back, and of course, most importantly of all, the length of time of survival of the patient, prognostic marker. Then we took it into a large cohort which had been collected from centres throughout the world. It was published earlier this year in Nature, called the Cancer Genome Atlas in ovarian cancer, where we were able to access that data where they also had linked survival with methylation, but using a quite different platform. And we were able to validate the observations that we'd made in the Scottish group in this large international study. And that's very important, obviously, being able to show that independent validation of biomarkers. One of the problems with biomarker studies, there's large numbers of studies, often very small studies that have not necessarily been properly validated. And if we want to use these markers as robust clinical markers that we can really trust, then it's really important that we're able to validate them across multiple sample sets. And so that gave us a lot of confidence in the data that we were seeing in the Wnt pathway. And I think really interestingly also gave us some insight into key genes that may be involved in this Wnt pathway in ovarian cancer. So what we're now doing is kind of going back into the lab to try and understand more about the mechanisms involved with these key genes and really trying to look at whether drugs which will target the Wnt pathway maybe have activity in ovarian cancer. So it's been a nice example of kind of going from lab observations into a clinical study and then going from a clinical study back into the lab again.
Would you discuss the potential of this in targeted medicine for solid tumors, and specifically with ovarian cancer?
Sure. Well, that clearly is one of our long-term aims. There's now a lot of activity in trying to take stratified medicine approaches, trying to identify mutations in given genes that will allow us to target therapies to particular patient populations. Now, up till now, a lot of that's been mainly looking at gene mutation rather than DNA methylation. And so clearly with some of these emerging observations on the importance of DNA methylation, we now want to take a similar approach. And indeed, subsequently to having looked at the Wnt pathway, we've now taken a same approach to 10 different new pathways. Well, I say new pathways, they're of course pathways that people have worked on for many years, but now looking at them in the context of CpG island methylation, taking the same in silico, evaluation, first of all, in our own cohort from the Scottish Gynecological Clinical Trials Group, and then independent validation in this large Cancer Genome Atlas validation set. And from that, we've now seen a number of really interesting pathways emerge, pathways which I know are very relevant to some of the new therapies that are coming through in ovarian cancer. Pathways involved in defective homologous recombination, which I know are really important for these emerging so-called PARP inhibitors, inhibitors of DNA repair processes. And also pathways involved in tumor angiogenesis, such as the VEGFs, which we know are again very important for some of the new agents targeting tumor vascularization and angiogenesis. So again, our thoughts are that yes, we want biomarkers that will tell us about the existing therapies, but will also allow us to place the new molecularly targeted therapies within that context so that we can really start to stratify patients and really hopefully in the future do our trials in perhaps a more mechanism-based approach.
Dr. Brown, thank you so very much.
Pleasure.